Okay, so good
Hello, everyone.
afternoon. So let me introduce you, and then you can continue. Okay, so good afternoon, everyone. This is session 3 for day 1 of our ATAL FDB. And the next speaker for the day is Mr. Deepak Purushottam. He is actually working as senior scientist at Washington University and St. Louis, USA. He specializes in devising data policies to integrate and visualize multi-scale publicly hosted data. His research focuses on designing innovative visualization solutions that enable researchers to understand the complexities of bioscience through integrative analysis and disseminating complex research results in a visual format. Today's title of his talk is Integration and Visualization of Multimodal Multi-Scale Data in Bioinformatics. I hope the session will be really fruitful for all of us. And I request Mr. Deepak to please start his session. Over to you, sir.
Thank you so much, Dr. Chandra. Hello, everyone. I'm really happy to be here presenting on this topic today. And I really thank you, all of you, for being here. So my talk will approach, this lecture series is about drug development and drug discovery in general. And my talk today will approach this topic from the perspective of genomics, doing bioinformatics at scale, and trying to integrate orthogonal data sources and what exactly is the intention there. So the drug discovery process in general, it can be mainly broken. You can think of, so it starts from target identification, advancing drug leads to candidates for preclinical and clinical studies. So all of this can be facilitated by genetic and genomic knowledge to some extent. And of late, bulk and single cell gene expression analysis from various population genomic studies are increasingly being used for understanding the biological relevance of a drug target and how to prioritize those drug targets. So understanding this biological relevance can in turn be understood by a keen understanding of the underlying biological system. And ultimately, the common genomic technologies that's being increasingly deployed for this purpose should eventually get interpreted in a joint fashion. So let me touch upon different aspects of what I just introduced and tie it all together into towards the end of the talk, where I argue that integrative genomic approaches can actually be a very useful weapon in your arsenal throughout your drug discovery journey. So the first step would be to understand the existing landscape of, we can call it experiments of nature. Essentially, experiments of nature is like naturally occurring mutations in humans that affect the activity of a particular protein target or targets that can be used to estimate the probable efficacy and toxicity of a drug targeting protein. And also this can be used to this process of getting a holistic picture of what all naturally occurring mutations are there in humans. It can affect the activity of and how it affects the activity of a particular protein target. This understanding will determine the success in translating the variant discovery data into actionable information for drug development. So use of genomics in drug discovery is not new. Genomic data is routinely impacting target identification and drug development. And this table here describes what are the common queries, and what are the representative sources that came out of those efforts in a form of a publicly available resource that anyone can query and go about as they proceed through the different stages of drug discovery. Data from these sources do not exactly compete with each other, but they are deployed to provide additional evidence to corroborate or recapitulate the primary story and to explain the underlying phenomena. So it is more intended, more likely to be interpreted in a cohesive manner. Genetic information can also be used to as a support to during the approval process. So there have been a lot of population studies where revolving around the clinical trials to corroborate that the explained phenomena on how the drug works is actually corroborated with some additional genomic evidence. So for example, this table gives some of the examples. I've left the link to the actual paper, and that goes into much more detail. One of the top examples here is about it's related with associated with increased cardiovascular risk. Basically, sequencing efforts of individuals with the loss of genes. The top three being the PCSK9, NPC1L1 or LPA. So the loss of gene or loss of function on these genes, those individuals were protected from disease naturally, while gain of function variants were associated with increased cardiovascular risk. So when we say genomics or genomic techniques, what are we talking about? Broadly, we can classify them into traditionally there have been lots and lots of GWAS and linkage analysis. So linkage analysis is basically these are association studies that use high density genotyping of common variants. So common variants being between 1 to 5% of allele frequency in a given population. Next, there is exome sequencing, and this is probably quite familiar to everyone. It just captures the coding sequences, which is around 1.5% of a human genome. And then, of course, there is whole genome sequencing. The recent trend has obviously been the cost has been coming down, so it's getting easier to do whole genome sequencing to get like a good quality coverage across the genome. Good quality would mean typically above 85%. And so whole genome would then also give information about the non-coding areas. So that is the expectation with a decent frequency. So this sequence coverage across all the regions, not just specifically exons, introns and intergenic region, all of this, whereas exome library will only lead to coverage across the targeted regions, typically corresponding to the exons. So on clinical grounds, whole genome sequencing is of particular interest for the study of rare genetic disorders that have no demonstrable finding after examining the coding regions. So that is one plus point for whole genome, but whole genome sequencing also comes with the added efforts needed to process that data, and just the discovery process is that much more complicated because of the additional moving parts. Uh, but so let's take an example of these profiling techniques that we are talking about, whether it's exome or whole genome. So tissue transcriptomics is probably really common now. So which basically is profiling of cells and tissues, um, and it supports the drug development process because a researcher can map the response to a given compound. Um, so this sort of interrogation of tissues and cells for the expression of a target of interest, and more recently, um, identification of causal variants associated with clinical phenotypes. Um, it's also increasingly being used as a really good tool to stratify the patients in clinical trials based on biomarkers that they could, any cohort specific biomarkers. So then focusing on some of the key elements of the non-coding genome that we would gain out of whole genome sequencing would be promoters, enhancers, insulators, or any other determinants that has an impact on the chromatin structure. Speaking of chromatin structure, which is a function of the 3D conformation of the nucleus. And as we all know, there is no fixed format. This is an evolving field, and a lot needs to be still understood about at least the last point. But because these tasks that we are listing down, this can be a behemoth of an effort, and it's not something that would ideally be done by a single lab. Although there are attempts to it, which leads to lots of medium powered studies. There is a constellation of medium power studies as compared to, say, more deeply powered and like with higher coverage. So for such intentions, um, most of the public funding is outlined to carry out research in a coordinated fashion where multiple labs join hands and try to tackle this problem, which is often spans into multiple years. And one such star example that we just for the at least for understanding the first 3 points, one such very well known consortium project which has spanned for almost 2 decades now, and it continues, is the ENCODE project. So for those of you who are not familiar with the ENCODE project, essentially ENCODE project was tasked, it initially started probably sometime shortly after human genome was sequenced, so probably around 2003. The goal of ENCODE is to build a comprehensive list of all the functional parts, all the functional elements in the human genome, including the elements that act at the protein and RNA level, and all the regulatory elements that control cells and circumstances in which a given gene could be active or not active. So ENCODE investigators are spread across the US and employ a whole array of methods to identify these functional elements. The discovery and annotation of gene elements is accomplished primarily by sequencing a diverse range of RNA sources, employing a lot of comparative genomics, integrative bioinformatic methods, and also it's a human in loop process. So human curators guide this effort. So the regulatory elements are typically investigated through lots across different assays. It could be DNA-seq, DNA hypersensitivity, assays to check DNA methylation, different immunoprecipitation techniques where you pull down the proteins that interact with the DNA and RNA, or histone modification, transcription factors, chromatin regulators, RNA binding proteins, all of these. There's a probably you can think of an assay. They're probably doing it and making this data available to the public. And then in the previous slide, I mentioned about 3D conformation of genome. Now that alone is like an effort that might quite well take like a few decades from now. So the most prominent consortium right now, at least in the US, is the 4D Nucleum program. It started approximately 5 to 6 years ago, and it's basically the intention is to understand how the nucleus is organized. What are the rules? What are the key players? How do they participate? What's the dynamics between those key players? Because ultimately all of this affects the underlying biology. And if we understand the biological system, we are paving the way, we are setting the stage to come up with mechanisms where ultimately when we have a query, when we ask a question, when the focus is around a given target or set of targets, or to see what's going around those targets, we have a platform that can answer those queries with acceptable confidence. So 4D Nucleum program is a brilliant example where the biological interpretation of interpretation is compounded when different data types are integrated, and continues to challenge us to reimagine how certain type of data type can be analyzed, can be visualized effectively. So I briefly alluded to tissue transcriptomics. There could be cases where tissue transcriptomics could get impaired by cell type heterogeneity. So this is a very common example that can be used to illustrate this aspect, that it's like taking a bunch of fruits. One way is to enjoy each fruit individually, and the other way is to just put it in a blender. And what comes out is basically this soup. And we then examine that, we run these sequencing assays on that soup. So we call that bulk RNA-seq. Whereas if you pick one cell at a time and just look at the transcriptome profiling, it is called single cell RNA-seq. This is probably quite familiar to all of you. But there could be cases where single cell might be the way to go, because single cell technologies allow direct measurement of and also it neatly lays out the differences between different tissues at single cell resolution, and you can do that for numerous samples, whereas you lose that power when we do bulk analysis. Now this is extremely case-to-case basis. There might be cases where bulk RNA-seq is perfectly all right, because we are now talking about, we are comparing liver with heart tissue. So we know that we are comparing like one class of cells with another class of cells. So if I get a readout which is indicative of a population of cells, that's fine. But for more nuanced questions, maybe bulk RNA-seq is not the answer, because there is an averaging effect, especially if you're going after rare variants. So, but there are so many cells. How I just mentioned that it would be nice if we have a platform where you could reliably perform queries. So there are efforts worldwide, and I'll list some of the prominent ones. Uh, so this is by the Human Cell Atlas. It is by the Chan Zuckerberg Initiative, one of the more ambitious projects at that. In fact, uh, so they are building a collection of high quality single cell atlas. It's they call it atlas because it it is from different organs. It's just not just one kind of cells. It's from different organs across the human body. So the goal is that this will result in a shared reference map, if you will, of the entire human body, which then sets the stage uh, to build platforms. It acts as the backbone to query expression patterns of different targets of interests. You can go down, you can start from the organ level, go to the tissue level, go down to the cell level, and perform very high resolution queries. So we are mostly, we touched upon different uh, genomic techniques, and we talked about one aspect, which is uh, any determinants or inhibitors of chromatin state, which means that, which begs the question that if we have the same copy of genome in all our cells, how is it that heart cell is so different in what it does and looks, and it's so different from, say, a liver cell or skin cell? So that brings me to the next part of what I want to talk about today um, is epigenetics. So epigenetics, if in case if you already know about it, then we can appreciate how epigenetics is something that is also when we are trying to understand, get a holistic picture. Now, of course, there are a lot of other ways to do this, proteomics, metabolomics, but as we expand our exploratory landscape, starting from genetics and genomics and focusing on transcriptomics, the next part is like there are consortiums to do single cell, there are consortiums to do understand the chromatin structure. One of the prominent fields is epigenetics, and I'll give like a quick primer to explain how vital epigenetics is and how it fits into the whole journey of drug discovery. So in a nutshell, epigenetics is the study of how DNA interacts with a multitude of small molecules present within the cell. Now, these molecules are, they don't really have, it just exists in the cells, and because of their presence, they act as, they function as a determinant of the chromatin state, which translates into the fact that they can activate or deactivate genes. And that cannot be ignored, because now our queries, our perturbations are, there is a new player which we have to factor in when we are asking questions, because the epigenetic state of the cell could actually be have a significant contribution in the process. So any cell will have an epigenetic state by virtue of mere presence and the concentration of those small molecules inside those cells. That concentration, that presence or absence makes all the difference. So like just to give like a super basic primer, we all know about transcription. We would expect the transcription machinery to produce the protein. The proteins are what defines cells, and gives all the characteristics and functions to those cells. But epigenetic states can change, and epigenetic states can boost or interfere with the transcription of specific genes. Now, this is a really curious phenomenon, but it is governed by very simple rules. And these rules play with each other. At a fundamental level, DNA is just to give it the structural the shape, the integrity. It is super coiled, and there are proteins that like we just discussed. Protein gives a characteristic to the cell. So one of the vital function is to keeping the DNA wrapped up in a configuration. So you can think of these guys which undergo like a systematic ordered coiling and packaging, but then these proteins can get labeled with small chemical tags, and these tags, the sum total of all these tags that associated with a given cell is called epigenome. This is like overly simplistic, but just to give like a brief background on what epigenetics is. So taking an example of a methyl group represses gene expression, and DNA methylation at promoter regions is generally associated with silencing. Now this sounds quite interruptive, but it might not be. Like we just discussed, we have the same genome copy, and the same genome copy has to work as a heart cell. It has to give instructions to the heart cell, and very different instructions to inside the liver cell. So maybe this is the inbuilt mechanism to silence the regions that does not really apply when we are in the liver cell. So the aspects of heart cell don't apply. So it has to be silenced in a systematic manner. So this is how nature found its way to do that. So more specifically, it just makes that region inaccessible. So it just completely shuts down all the pathways of those genes. So gene is still there. It's embedded inside, but now it doesn't have access to any of the transcription machinery. So it's just physically taken off and buried, tucked away into a region where it cannot accidentally. But if this thing were to happen in, so for example, like I said, DNA methylation at promoter regions is generally associated with gene silencing, and it shuts down the related pathways. But what if the pathway we are talking about is concerned with DNA repair? That DNA repair would normally help prevent tumor from proliferating, tumor cells from growing and proliferating. And if the DNA repair pathways are silenced, that just leads to a really unfortunate situation, fortunate for the cell which is off the hook and trying to grow. So um, it's important to then start understanding that can these mechanisms which work in binary states, or they work in an orchestrated fashion with other players, can things go wrong? And if things go wrong, can we account for it? Can we profile it? So essentially, just like how we talked about tissue transcriptomic profiling, we can do tissue epigenomic profiling, and we can now look for patterns of uh, expression levels of an epigenetic protein, and then look for substantial differences between the disease state compared with the normal physiology. So this is just to summarize that the DNA is very much there. It just happens to be in the region which gets tucked in, so it shuts down the pathways. Of course, if it's important enough, cells have redundant pathways, so multiple pathways lead to the same gene product, but then that gene product, the concentration might fluctuate. That fluctuation could lead to a situation which, when extended chronically, might lead to a disease state, or a precursor, or one of the predisposition towards the disease state. So that was a quick primer on how the epigenetic reprogramming is what is largely responsible for some of the cells that develop into heart versus some developing into liver cells. And one important thing to note is the epigenetics and epigenomics undergoes transformation throughout our lifetime. So environmental variables, environmental toxins, environmental factors, if you will, can modify and affect individuals. So identical twins can grow up to be completely different individuals. Now, of course, there is an aspect of nature versus nature, um, uh, but epigenetics do modify and lead to different phenotypic states, which can culminate to a diseased or healthy state. Now, this means that epigenetics can be exploited to, if understood, it can be exploited to reverse this process, reverse these changes that led to diseased state. So again, going back to the central theme of what I'm trying to convey is knowing and learning about all the moving parts of the biological system adds a layer of understanding and adds a layer of things that can factor what we are ultimately trying to achieve with the desired target in mind. And uh, if a region is methylated, we use some chemical to demethylate that region in a very specific, precise manner. So that could be a drug intervention strategy. In fact, this is very much an existing field. It's fast evolving, and there are many drugs which have already come out of this process. This is not new. And so in summary, a complete understanding of disease associated epigenetic abnormalities, and also if we understand the somatic mutations involving epigenetic proteins, with, say, resulting in gain of function or loss of function that predispose towards disease, if that can be identified, those become a sort of a meta target, because now we can see what are the pathways that they participate of our, so we they could be used as a disease associated epigenetic abnormalities can be used as a good proxy. So each of our 200 cell types in our body essentially has it essentially has the same genome copy, but it's distinct epigenome. So now it would be obviously an enormous task to actually go through the process of characterization of each of these cell types and summarize the epigenomic signatures, and build some sort of a atlas or a compendium. But that arduous process is what paves the way for setting a mechanism to compare few cell types of interest and detect druggable targets. So one analogy that I really like is to remember what epigenetics is. To if your genome is the hardware, epigenetics is the software. So the same copy goes into every cell, but it acts quite differently. So this is a cool analogy. Speaking of profiling 200 cell types, as you can guess by now, there must be some monstrous undertaking which is publicly funded and trying to do it at an enormous scale. And that is absolutely right. And that consortium is called Roadmap Epigenomics. And our lab was actually quite fortunate to be part of it. It started almost a decade ago. And the goal was to define the first complete reference. Like we have reference genome, we map the profiled sequencing read against the reference genome. So the goal is to build like a reference epigenome of all the human cell types, and to complete around 25 high quality epigenomes. So this was an NIH-led effort. The goal with, like with any public funding, is to produce a public resource of data that can basically catalyze biology and disease-oriented research. Of course, it assayed a whole bunch of different epigenetic related assays, DNA methylation, histone modification, chromatin accessibility, small RNA transcripts, frequently involved in human disease state. So the consortium closed the gap between data generation, and also led to an important direction, which is the public dissemination of and the rapid release of all this data, the epigenomic profiles, the highly integrated maps. How do we release this, disseminate to the scientific community? So this goal led to the development and standardization of some of the dissemination protocols amongst a whole other set of setting best practices. So it was it started out by in the first 6 years, around complete 100 epigenomes were sequenced, and there were a ton of partially completed epigenome spanning across diverse sets of tissues and diverse sets of epigenomic assays. So when what you end up with is a huge compendium of these results across cell types. So is um, this is gold for a data miner, for a researcher who is trying to who is working on a target gene, and now they have this free public resource they can consume to see how it strengthens their biological story. So yeah, this is just a representation to show the magnitude of how lots and lots of data sets were generated across different organs, different cell types, different assays, and different conditions. So this map continues to evolve. It's not exactly linear. There are many dimensions to this atlas. Now I have, at this point, I have talked about a few large publicly funded consortiums. So what happens to all that data? How do we are talking about target of interest, but how exactly do we consume that? So I just talked about the dissemination into scientific community. How exactly does that happen? So that brings me to the last part of what I want to discuss today is how do we integrate and visualize these different modalities and the data that could be of different scales. You are talking probably one is at like a whole genome level, and some is something very targeted. So there needs to be some grammar to integrate all this and to normalize it and to give us confidence that what we are looking is okay quality. This is how it should be. Now once you have that baseline confidence, then we can rely on the results we are looking at more confidently. So let me illustrate this with one example. So all these consortiums, wouldn't it be neat that if we could just have like an application where we could just select that today I want to consume these data sets. Now I just want to put it in this session. I just cherry pick all different data from different consortiums. I have some query in my mind, so I'll just get the data, just put it in some in one centralized place, and then I obviously have a target of interest. So I want to go straight to the target, and so maybe it's a gene, it's your favorite gene. So this is just a representation of that gene, and it sits on chromosome 10. The gene feature starts from so and so coordinate and goes all the way to so and so coordinate. Now, just for that gene, I want to layer, just like layers of, say, layers of an onion. I just want to stack my RNA-seq profiling results just for that gene. And I have 3 different conditions. It could be 3 different conditions, maybe 3 different cell types, or 3 different replicates, whatever. I have 3 different data sources that I want to look at. It would be neat if I could just color code it. Now, the problem is I also happen to have some more data, which is I want to look at some methylation data, because transcriptomic information was good. I want to add a layer of epigenetics and see how, for this region, how do things stack up. So we are entering the territory of doing an orthogonal step through of bringing different modalities to one place, stack it right against the target of interest. This is getting interesting. Now let's go one step further. We want to add ATAC-seq. Now ATAC-seq, if for those of you who do not know, ATAC-seq is an excellent asset that shows the chromatin accessibility, general chromatin accessibility state. It is a very good indicator to see how, for that snapshot, how accessible or not accessible the chromatin area was. So I want to stack it up again. I have color coded it in a similar fashion. So whatever those three conditions were or cell types were. So then on the exonic region, I spot some interesting trends. It's interesting because I see something that I was expecting, and it all matches. Like I see some expression, and the methylation matches up. Biologically, it makes sense, and it also happens to be it happens to be accessible, because if it's accessible, only then the transcription machinery would have gotten access to that region and spit out products of transcription. So but it would be nice if so this is the end goal. So for any given region of interest, you can ask biologically relevant questions. For example, it is known that promoters typically have low methylation. So so the track names which have bluish background, those are from one specific cell type. So like in this case, oligodendrocyte. So I'm taking oligodendrocyte RNA, stacking it with methylation, oligodendrocyte methylation, stacking it with oligodendrocyte ATAC-seq. So what I see is right on the promoter region, the methylation is low, as I would expect. And the accessibility is high. So this gives me, so I can do some more checks. I can use some biological trends to see things. Now it's important that you have stacked it up, but these could have come from different assays. So how do you know that there is there is no mistake? Like maybe the trimming was not proper, so all the data is like off by a few bases. How do you line things up? So you look at these biological landmarks and use that to drive home a point that, okay, you add a layer of CpG islands. For CpG islands generally lack methylation, so you would expect CpG islands to be hypermethylated, and majority of CpG islands are associated with in the neighborhood of gene promoters, because that sort of goes with the there could be some biological reasoning. So with using this information, which is like a general biological rule, you can then see that things line up, everything looks proper, things are not off by a few bases. So it acts as a QC check. So you could use a few different scenarios to line things up, and once this is done, it gives a high confidence to believe the trends that we actually wanted to see, which is that I wanted to compare oligodendrocyte with motor neuron, and I wanted to compare the methylation level for the given target. So I see that oligodendrocyte is less than astrocyte, less than motor neuron. So this result is highly believable. This is we followed some steps to ensure that we can minimize the randomness as much as possible. So due to the steps that we took initially to integrate, to stack things up, then line things up, um, against that backdrop, we see a result, we spot a trend which gives us more confidence, and then uh, it corroborates with the ultimate pattern. So this now wouldn't it be nice if what we are looking at, if this was uh, a web-based software, so anyone could use it because the data is public. You could import the data, you could just do your analysis, and now you want to share it with a collaborator, so you can just save that session and it generates a URL, and you could just share it with your collaborator and they can load, and in one picture you tell the whole story. So this is a simple example, but there are lots and lots of very detailed mechanisms, and this platform that I'm discussing is something that I work on. This has been in many years into development. It's called the Washup Epigenome Browser. So it was like first started more than a decade ago, and it was uh, there are a lot of genome browsers, but this browser was specifically uh, geared towards um, the the epigenomic space. And like I mentioned, roadmap epigenomic project was uh, the project which gave the biggest impetus in moving things forward, and this became the de facto dissemination and distribution platform for uh, that consortium. And then instead of we ask why wait, like we can use this open platform to also distribute other public data. So in one place you get all these different public data hubs. um, so this is the URL to access this browser, um, and I can just quickly just talk about the layout on what essentially a browser is. A genome browser is just like how you are basically using Google Maps to go from one place to another. So, or you want to look at a neighborhood of what things are in this neighborhood. So it's like that. So you have some annotations on the top which define the region that you're looking at. Chromosome 10 starting from this base to this base, spanning across so and so kilobases. Um, and then it shows all the different tracks that you have stacked. Now each track is like a unit of data which is a product from an experiment. All right. And the neat thing about this software is we can zoom in and zoom out and scroll and just go from one region to another. You can put the coordinate of your region interest. So it makes it really easy to just focus on targets of interest. Although you could theoretically hop from one place to another. And there are ways to easily search and add tracks to this browser session. So in one session, you can filter a whole bunch of different kinds of data and add it to the same session. Use different configuration options to customize how things look. You can change the height of, so whole lot of customizations. Really feature rich for a web application. It comes with um, a slew of features that can be used to customize the tracks to make it look make the at most biological sense. For example, you want y-axis to be normalized uh, against all the tracks, across all the tracks. So otherwise some peak might look like way high, but then it is not really that high. So you could normalize those things, use the configuration settings to easily organize the tracks. So for example, in this example, we have taken a whole bunch of astrocytes and put the background as red, so it is visually more nice and clear. So uh, we could add any number of tracks like this, and basically we are generating a picture worth 1,000 words. So this is obviously geared towards generating really high quality publication figures, which then can be used to drive home the point when sharing research results. Yeah, this is another example where um, the background is coded by male female, so as to show the trend easily. So you don't really have to like, if you have one target, what if you have 5 targets? What happens if you have 15 targets? uh, there is a neat feature which is called as region set view. So you can just make a list of all those gene coordinates, and it just puts those regions, it juxtaposes it right next to each other. So this is one of the unique features. There are quick summarization tools, like making histograms, scatter plots across a region set, uh, and all of what I'm mentioning, it's like very nicely described in the documentation. So for those of you who are curious about this, just go to the URL that I mentioned before. I'll again post in the chat here. Um, so this brings me to the question: how is this happening, man? And if it's a web-based software, obviously we can't load all this data in a web browser. Obviously, there must be some sort of complicated mechanism to host this, and obviously it is not scalable, and everyday biologists can't really make use of this. That would be incorrect. It's actually quite simple, because it uses the configuration is all in this simple. Uh, um, it's the file format is called JSON, and you can describe what track, and you can describe all the metadata that goes with that track, and um, data specifically. If you have your own data, if you want to use your own data against the backdrop of the public data, there is a data preparation protocol. So you run a couple of commands. Basically, what's happening is you're formatting and indexing and compressing that data, and you upload it to a web app accessible location. If this is a big problem, you can contact us, and we can help out with this aspect. But data lives in a web accessible location, and when you query with a target of interest, um, it specifically it uses that index to just get that specific slice, and then it streams it back to the browser and just loads that specific view. So the actual data could be like in GBs, multiple GBs, but it still works fine, because at a given time we are only looking at a few slices of data, and that few slices of data across different data sets. So that's the beauty of this. um, uh, this mechanism is not something that we invented. It was invented by UCSC Genome Browser, which is prominently arguably one of the most popular genome browsers right now. uh, and the Epigenome browser falls into a niche where it really evolved around providing more specialized epigenomic based analysis functions. Um, so for those of you who are more curious on what you're looking at about the data retrieval part, um, so most recently we published in 2019. So we have described how this works under the hood and how it can stream all that data straight to your browser and draw all this um, quite swiftly. Um, so it doesn't really need a server or anything. So you could just open the URL and just start a session, just start importing some of the public data in your off the basis. This platform can be extended to build more specific functions, and I'll give an example of what some of the work we did uh, over the past year, especially in the context of the pandemic. So so this can be extended to analyze different SARS-CoV-2 strains. And one of the examples is so on the left side, the zoomed in view, the orange box, that's displaying the first 9 amino acids of the SARS-CoV-2 N protein. And so there is a SNV, single nucleotide variation, which goes T to C at 28,296 base pair, the position, and then at 28,299 there is a G2A SNV. But these are silent mutations. However, at 28,294 there is a TCA insertion, and that so that insertion is in the first strain, the one that starts with AY. This insertion actually results in a serine residue in the SARS-CoV N protein as compared to the CoV-2 N protein. So and one point to note is this is not present in other CoV-2 specific strains. So the bottommost track we are looking at is putative immune epitope information. Now that information is integrated from an open database. So there is a database which IDEB, so that that database has a lot of, it's a great resource for putative immunoepitopes. So we can add a track which for this specific region, for you can query the virus, the strain, it will give those putative epitope. And because of what we just discussed, this putative epitope is probably not a great candidate for vaccine design. So just to summarize what we just did here, we looked at the single nucleotide variants, we looked at indels, and then we came to a conclusion that when you're designing an epitope driven vaccine, you would want it to have maximum efficacy, and also if it is consistent across all the known variants, there is a good chance to go after it. For example, the next example that I show, so the left box, if you look at the zoomed in view, the purple box, and we are looking at the zoomed in view on the right side. So it's everything is quite similar. We're looking at the region, but here we don't see any variation. So this may be a good candidate for epitope design, because it's fully conserved across the SARS-CoV as well as the CoV-2 strains. The first two are SARS-CoV, and the last 5 strains are CoV-2 strains. So yeah, so it encodes yeah, so this is an example of how this approach can be used to carry out this kind of analysis, where you're layering different kinds of data, uh, then making sure that if it's publicly hosted data, it's all quality controlled before becoming a public resource, so we don't really have to worry about it. But in case it's something that you generated in your own lab and you want it to assess it, the quality of it against the backdrop of the wealth of publicly available data sets, this tool makes it really easy. So I would encourage, if you guys are curious about this, please go ahead and let me go back to that slide which had the URL one more time. Yep. So it is epigenomgateway.postal.edu. All right, now I'll quickly wrap it up with some of the last things I want to discuss here. We are talking about web-based strategies to prepare, host, integrate, and visualize data. This process cannot be complete without touching upon some of the more recent trends, and I'll just briefly discuss, because this could be like, this doesn't exactly apply to drug discovery, but it could be something where it could be something that it could be good to know, and especially all the large data consortiums are gearing towards this direction. So the central objective is this. Throughout this talk, I mentioned several times like how we have so many data sets to profile, and all of this is located somewhere. So if we were to uh, download all this information, download all this data to one place to run some bioinformatic pipeline, it would be uh, all right, and that's the traditional approach. So the traditional approach is bring data to researchers. So so unlike the traditional high performance clusters, for which you tend to script your analysis in a way that's very much dependent on the environment, the model presented in figure really encourages thinking about the portability of your analysis. So if we can make our analysis portable, instead of bringing the data to researchers, we can take the analysis and the tools to the data. So it's like inverting the paradigm. So with multiple cloud offerings now, they're all vying for market share, each storing and providing access to multiple data sets. Researchers today are going to want to apply their algorithms to the data wherever it resides. So highly portable workflow languages that can run in different systems on different clouds have become popular over recent years. So what's the downside? Downside is obviously the move to the cloud. It adds a whole new layer of complexity, or possibly several layers of complexity to already non-trivial world of research computing. So although some researchers might already have sufficient training or personal resources to figure out how use cloud services effectively in their work. But undoubtedly, that's the minority right now. Much larger majority of biomedical research community is generally not equipped to deal with this kind of with like bare metal services provided by public cloud vendors. So there is obviously an urgent need for development of platforms and interfaces tailored to the needs of researchers that abstract away the operational details and allow those researchers to focus on their science. So I just wanted to bring this up because to some extent the tool that I just talked about sort of follows that paradigm. It takes it loads that integration and visualization process in the client's browser. So there is no download, just the required data is streamed to just do those calculations to generate those visualizations as needed. And also there is a lot of push, there is a lot of impetus to especially in the context of these public consortiums to follow the fair data principles. So FAIR stands for findable, accessible, interoperable, and reusable. So extending on what I mentioned earlier, generalizing the format, the workflows, porting it into a general purpose format which the entire community agrees upon, it brings a lot of benefits. So now you might think that you're going after a target of interest, and you would be curious, like, let me see what else is going on around this space, who has worked on this. Now, if only it was easily findable, if you could access it, and then it was in some sort of interoperable format, you could reuse it. And that saves a lot of time. And that data which was generated once ends up getting used by a completely unrelated researcher across the world. So there is a big push to do science in this new the obviously this requires us more effort upfront, but that's for the community to come together and work through those muddy details. But once those details get figured out, it's gets easier to implement and then repurpose it as between one collaboration and other consortium. And one of the other recent trends is that it is not just a data consortium anymore. Things are gearing towards data ecosystems. So you would think like there are different data consortiums that can talk to each other. You don't really need to import the whole metadata all over again. You're working with an interoperable file format, so you could theoretically just pick any data across the different consortium, host a shared space on the cloud, a work session, bring in all this data, and start a job. Now, this would obviously be incomplete if we don't discuss just the core nuggets that you have different levels of compute, and cluster is often used for more long running jobs which are highly now I agree this might not apply for a lot of desktop oriented softwares, but I just wanted to share a general sense of how workflows are now getting packaged into an interoperable file format. And interoperable is an easier word to say. How do we accomplish it? So one of the more popular paradigms is obviously by now you guys might know how when you run something on your physical machine, you're sort of managing the software, the dependency, the OS. Um, but but then there are concepts like virtual machines and containers. So some of you might have used both of them, which essentially uses the same infrastructure but hosts different guest operating systems. So the apps could run in their own silo, as if that app thinks that it's the only thing running on the OS, so it has full control on the resources. Um, the more neat way to do this now is to use Docker. Uh, so just like the hypervisor in the virtual machine setup, Docker daemon sort of becomes like a common denominator, and now any pipeline can be compiled into a Docker, and that Docker will, if it works on your laptop, it will work on any cloud across any cluster. So that's how you make your analysis, your workflow. So this is one example where you could use some of the components and port it over to more generic formats, interoperable formats, and then run the same. It automatically splits it into individual chunks, and based on the requirement of compute, it will automatically allocate to those tasks and work on the cloud and send back the results. These these techniques might not apply to a lot of softwares right now, but I just want to underscore that these are more recent trends, and more data consortiums are trying to go in this direction, because it sort of fulfills the philosophy that I mentioned earlier of inverting the control, going from bringing the data to the researcher, and inverting that model and taking the researcher's workflow, packaging it into an interoperable format, so now it can run on any cloud. And I would end with just dropping, there are different tools to now interact with the data directly. A lot of like how I mentioned the Epigenome browser. Similarly, you must have heard of Jupyter notebooks, which are getting really popular. So the basic idea is to combine analysis methods and findings in a single place, in a form that that anyone can easily distribute, in a way that this is a logical evolution of traditional scientific paper, if you will, but much better, because it dramatically shortens the path between reading how an analysis was done and actually being able to reproduce it. It's difficult to overstate how powerful this concept is, and what a dramatic impact it can have on the reusability and reproducibility of findings in the computational sciences. Yeah, that was my last slide. I'll stop now. I'll be happy to take any questions.
Okay, thank you so much, sir. Audience, you can ask questions. Yes, you can unmute yourself, or you can put your questions in the chat.
Sir, I have one query. Can I audible?
Yes,
Yes.
I can hear you.
Can we directly find the epitope in this online server, Epigenetic database?
Not on this server. The epitopes are in the IEDB software, IEDB database.
Okay.
And this specific example makes use of one of the file formats to bring it in a format that can be imported in this website. But IEDB is the platform, is the database where you will find all the epitopes.
The another question related to this epigenetics. What kind of behavior in human can affect the epigenetic switches?
Sorry, come again.
What kind of behavior in humans can affect the epigenetic switches or epigenomics?
I see. That's a really interesting question, and something which is being studied a lot. To my knowledge, I can think of two things. One is definitely the immediate environmental factors, and that could mean any kinds of stressors. It could be either some environmental toxin, or there have been recent studies, not on humans, on mice, where they were able to um, actually show that um, the mother mice, when it ignores the pup, uh, when it's calling for help, uh, it sort of led to, when the mice grows up, it sort of led to stress induced epigenetic changes. So these studies are obviously quite difficult to carry out on humans. So mice is a proxy model. So a lifetime of human is roughly 10 months in mice's life. So a lot of these perturbations are actually carried out in mice. There are a lot of mice studies going on. So those results are used as a proxy to extrapolate and come up with like an estimate of how it would impact humans.
Okay. Any bioactive, or we can say the genetic modified food can alter the DNA methylation? Any study in your lab, like?
I have not come across any study specifically on that topic, but that will be something to keep an eye out on.
Okay, fine.
Sir, how is this genetically modified food, they are harmful for us?
Sorry, what is the question?
My question is that genetically modified food is harmful for us. So what can we do? We have come in contact with so many food we are taking nowadays.
Yeah,
Because
unfortunately, these will be something studies have to present, and this is like a complex space to come up with like a clear understanding of mechanism. The best we have, like I mentioned, is mostly mice studies. So I would assume taking a look at a lot of epigenetic therapy. If you search for epigenetic therapy, the most recent trends, there are studies coming out now which essentially are mostly centered around curing cancer and more severe diseases, which can be well characterized. So there is like a target to go after, and by making those epigenetic modifications, there are recent studies where they are able to cure blindness, and so epigenetics alterations combined with CRISPR-Cas9 methodologies is something that's being pursued quite actively. And I'm sorry, I don't exactly know anything specific to your question.
thank you, sir.
There's a question in the chat box. I just read it.
Hello, sir. Am I audible?
Yes.
Yeah. The basic Python is enough for to simulate like this, or we need to study for BioPython?
It might be quite specific to what kind of analysis you're doing. I would assume. I mean, to answer your question more, I can answer that in a more generalized way. Any pipeline which is probably authored by the lab, they might use some of the mature software packages to get the job done. But in more recent months, maybe there is a better package which works better, or things like that. It's a very subjective question. It goes case to case basis, very specific to and of course there are multiple ways to do the exact same pipeline. So I assume BioPython might be factoring in some of the developments and making it easier for biologists to start using more, just removing layers of complexity. But specifically to your specific analysis, there might be a chance that the latest package doesn't work because it is maybe still on Python 2.7 as compared to say Python 3.8, and the BioPython package that you just installed, it's complaining that it doesn't like one of the other dependencies. So it's hard to really understand. It's probably best to just give it a try. And any pipeline, you should probably contact the author and ask for some help if you run into some installation troubles. Yeah.
Okay, we can take just a last question now. Anyone wants to ask? Any more question?
Hello.
Yes.
Dr. Deepak Purushottam. This is Narendra. Thank you for the great presentation. I was checking browser for analysis purpose, and I wanted to see if there is some help is available how to use initially. And there was a YouTube link, but there is no information over there. Could you help me how to initially, if someone wants to use this browser for,
Sure.
analysis purpose?
Sure. So if you let me so on the website, if you go, you'll find a link called documentation, and let me try to quickly show it here.
Sir, give the example of first one, human HG19.
Sure.
That we will get idea directly.
Okay, sure. Do you see my screen now?
Yes.
Okay, so the easiest way is to, when you go to this browser, explore the new browser, and here on the right side you'll see documentation. So if you click on this, it will walk through all possible combinations. You can just start from use the browser, and it will just give you all the examples on how to understand this, how this UI is laid out.
Thank you.
Yeah.
Okay, I guess we have come up to the last end of this session, and I would like to invite Dr. Rashmi Singh to please extend a vote of thanks to Mr. Deepak.
Thank you very much, ma'am, for giving me this opportunity. So thank you very much, sir, for this enlightening presentation. Your presentation not only informed us, but it also inspired and motivated us. It was very interesting to learn about different genomic techniques, 4D nucleome program, tissue transcriptomics, human cell atlas, and different aspects related to epigenetics and epigenomics. So thank you, sir, for accepting our invitation, and thank you very much for this wonderful presentation. Thank you, sir.
Thank you so much. Thanks for your kind words. I truly appreciate this opportunity, and I thanks all the participants for your time and attention. Thank you so much.
It was a very informative talk, and I guess the time was less, and we need to have a big sessions for your training, because that was a lot of information, which I am also not aware about a lot of things. I'm into bioinformatics itself, but there are a lot of things I'm not aware about, the softwares and whatever things you were just telling us. So I hope so we can have some future workshops with you. Okay.
Absolutely.
Thank
I
you
just
so
wanted
much.
I just wanted to add that I'm myself an alumni of IILM. I passed. I did BTech in biotechnology. I graduated in 2007.
Yes, that I'm aware, and it's really it's an honor for every one of us to know about this, and It's really good to see our own alumni to go out and have so much good hand in research in bioinformatics and genomics and all these fields. So we are looking forward for more sessions for faculty as well as students. It's a never-ending learning process for all of us. Okay. So thank you once again to all the participants, and we'll just join the session tomorrow again at 10 a.m. So we'll continue with 3 sessions, 10, 12, and 2:30. And please do mark your attendance. I'm sharing the link on the chat box as well as the WhatsApp group. And if you're having any kind of query, then you can message me through WhatsApp. Okay. So thank you again. I'll be ending the session now. Thank you, everyone.